One of the things that Mendel, when he was studying the garden pea plant, and he looked at all the various characteristics, he discovered some patterns. And one of the patterns that he discovered is that for whatever trait I'm talking about, and the characteristics he had there were quite a few, like this, how tall the plant was, what color was the seed coat, was the seed coat smooth or wrinkled, what color was the seed pod. I mean, he did all kinds of stuff. Well, he noticed that each characteristic is controlled by a pair of factors, is what he called them. Today we call them what? We call them genes today. Okay? Now, these genes can be either dominant or recessive. Okay, these genes can be dominant or recessive. Doesn't mean that you have to have both necessarily, but that's the possibilities. Dominant means it's a trait that if two opposing traits are there, it will be the one that shows up. Now, what we, in our pedigree, human pedigree, we're doing the one for about earlobes. So an unattached earlobe is a dominant trait. Why? Because I look around the room and more of you have this than anything else. Okay? So it's typical of a dominant trait. That means the one that shows up most often. The recessive trait is hidden and only can show up under certain circumstances that we'll cover in a little bit. So if you have attached earlobe, then there are very few of those, and that's the reason why is they're usually masked by the dominant trait. So if you have a dominant gene and a recessive gene, the dominant one's going to cover up or mask the other one. You won't see the other one. When it comes to representing these genes with letters, which you have to do on your assignment on question 6, it says you're going to use heterozygous, and we'll figure out what that means in a moment, but we can pick whatever letters we want. They don't say in the question what trait that you're talking about, so you could pick any letter that you wanted. So typically what we do is we use capital letters for the dominant trait. So in other words, we usually take the dominant characteristic, whatever that is. If it's like tall, well, then I'll use a capital T for tall. And then if the recessive trait then is short, I'm going to use a lowercase t. So we use a capital letter for the dominant trait, and we'll use its lowercase version for its recessive trait. But it doesn't really matter what letters you pick, just some letters make a lot more sense. If you're talking about, tall and short, capital T for tall sounds good. If you're talking about yellow and green, and yellow is dominant, I'd pick capital Y. So it doesn't really matter what letters you pick. Now, when it comes to forming sex cells, like in humans forming sperm and egg, in the process of forming that during meiosis, that the pair of genes segregate or separate and go to separate sex cells. In other words, if you possess, let's say, a capital T and a lowercase t, one sex cell would get the capital T, a different sex cell would get the lowercase t. They don't go to the same one. So that's what it means to segregate. They separate from each other in the formation of sex cells. So here would be an example. Let's say we're using height, and let's say this tall plant happens to have both genes for tall, both dominant genes. When it comes to forming sex cells, some sex cells will get this one, and some will get this one. So as far as the trait is concerned, or the kind of gene is concerned, they're both going to get the same kind, right? So there's not any difference between them as far as that's concerned. Now, let's say we have a short pea plant, and to be short, it has to be both lowercase. So when they form sex cells, it'll form, they'll go to separate places, but again, in that situation, we only really have one kind of sex cell as far as that trait is concerned. They're both getting the same thing. But when I put them together, one from the male and one from the female, or the female and the male, doesn't really matter which one's which. You're going to get one of these and one of these. So you could have capital T, capital T. So let's take this first one and this one, the first one and the last one. They're both capital T's, little t's, aren't they? Then if I take this capital T and this little t, I get this. But if I take this one and the last one, I get the same thing. So what do you notice about all of the offspring? They have all the same kind of gene combination. We call that genotype. They have the same genotype. So genotype is what letters you have, you represent with it, and they're all going to be that. So in this combination, they're all going to be tall, because as long as you have one dominant gene, it's going to mask any recessive traits, so all the plants are going to be tall. Now, we're going to talk about something where this will show up one more time. This is kind of like what's called a test cross. If those of you that have filled your notes out far enough, you know you talked about a test cross. That test cross is where you're crossing it with an organism that has the recessive trait, homozygous for the recessive trait. So here is one where, let's say we took two of the offspring from the first generation. Notice that their genes segregate. Now, what are the possible combinations? I could take this capital T and this capital T, and I get that. Or I could take this capital T and this little t, and I get that. Or I could take this little t and this capital T, and I get this one. We always write the capital letter first. You know, that's kind of like the customary way of doing it. There, we could take the last one and the last one, and we get this. So in this combination right here, I get how many different genotypes? How many different ones? How many different combinations of T's do I get? 3, right? I get one that's like this, 2 that's like this, and 1 that's like that. That's kind of like what they're asking about in number 6 question, except they don't want it drawn like this. They want it in the little box, you know, the Punnett square one. So let's define, make sure we understand the terms that I've been using some of these terms, but let's make sure we understand what they are. Phenotype means what does it look like? So since we were talking about plant height, is the plant tall or is it short? Okay, we're not asking what letters it has. We're asking what does it look like when I say phenotype? Or is it unattached or is it attached? That's what we mean by phenotype. What does it look like? Not what genes I have, but what does it look like? The genotype is what actual letters do I have. Okay. So on your human pedigree project that you're doing, that's one of the things I said I would elaborate on at a future time, that's now, is that in every one of those shapes, whether it's a square or a circle, you need to tell me, is it, is it both capital letters or capital little letter or 2 little letters, okay? Well, if it's unattached, which letter combination would you want? Now, since I'm using ears, I'd probably be attached and unattached. And since unattached is dominant, I'd probably use the letter U. But you can pick whatever letter you want. I don't care. Okay? You just have to tell me what letter means dominant and what letter means recessive. You could have a little key on the side that tells me what letter I'm using. And so if I'm like this, what genotypes would produce this kind of earlobe? What kind of genotypes would produce an unattached earlobe?
2 capitals.
2 capitals, or what?
Or 1 dominant.
Okay, 1 capital and 1 lowercase. Both would produce the same thing, right? So if in the process of you doing your pedigree lab, you don't know which one of the two it is, there's no other information that I can narrow it down to one or the other, then you put both in there. You could put down like, if you're using T's, it'd be capital T, or capital T, little t. But if I have an attached, it's pretty easy, isn't it? What will I write in that box?
Lowercase, lowercase.
Yeah, lowercase, right? And the ones that have attached earlobes. So I've already given you some ways of figuring that out, but it works best. I'll give you a hint on that human pedigree is work from the bottom up. Like you are the proofs that your parents had certain genotypes. And then your parents are the proofs that the grandparents had these genotypes. So it's the offspring that always kind of prove what the previous generation is. We don't know what you might be unless you have attached. Well, that's pretty easy. But if we figured out the parents, that this parent has to be homozygous dominant, in other words, they have both letters the same, then you can pretty well figure out what the next generation is. Because if that has to be that, then they can only give dominant genes. That parent can only give dominant genes. Yes.
So to put down a capital T and a lowercase t, would it have to be like one parent has attached and one parent has not attached?
Okay, so to rephrase the first part of your question.
Like to get the capital T and lowercase t.
Okay, for you to have the big T, little t, that means one parent would have to have at least one capital T, they could have two, and the other one has to have a lowercase t. Do we know what the other one is? If that parent has attached earlobes, then we know then it's both lowercase, right? If it's unattached, then they have one of each, right? But they got the lowercase, but we know they have to have one of each, otherwise there's no way that could have happened. So here's some other terms that we sometimes use in genetics. Locus, that's the specific site on a chromosome where a particular gene is located. So in other words, so they're saying a specific chromosome and where on this chromosome this gene is located. Last night on the news, they had a piece that they were talking about MRSA. I forget what the letters stand for, but it's a very kind of like deadly bacterial infection you don't want to get because it's a lot of strains of MRSA are antibiotic resistant. And there is a professor or a doctor at Creighton that has done research on that, and they have now narrowed it down to what letter. Remember when we talked about that 3 letters make up a gene? They figured out exactly which letter on this whole chromosome is the problem that's allowing that bacteria to change to become antibiotic resistant to whatever antibiotic that it's being exposed to. Try another antibiotic and it changes. So they figured out exactly what what letter in the gene is causing it. Now the goal is to figure out how can we keep that from changing? How can we maybe is there a medication or is there some medicine or something like that when exposed to this, it prevents the gene from being turned on or off. I don't know which way what the problem is. Is it turned off or on? Probably on. Maybe we can figure out a way that gene is always turned off. And then we actually can figure out a way to make sure that that's not a problem anymore. We figured out a way. So that's the work that's going on right now. And if some of you like that kind of work, is there any shortage of diseases that you can work on? No, there's diseases You know, for a whole lifetime to work on to figure out, okay, how can we, but remember, that's our creation mandate, is to make life better for people. And what better way to figure out a way to turn off a gene that's causing a lot of people to get sick, and with MRSA, there's a good chance you're going to die. So, so locus is where it's located, and allele is the alternate forms of the gene. So for instance, we're talking about height or earlobe. There's a gene for attached and a gene for unattached. Those are alleles. They're alternate forms of each other. Homozygous, the prefix homo means what? What's the prefix homo mean?
It's like other, right? It's like
It means the same. Okay? So that means both genes are the same. Either like both capitals or both lowercase. That would be homozygous. That's a possibility for both. Heterozygous means that you have difference. You have one capital and one lowercase. One capital, one lowercase. That's what it means for heterozygous. So you can have homozygous dominant, okay, and that means you have both capital letters. You can have homozygous recessive, where you have both the same, but they're both lowercase. Now, heterozygous means you just have to have one of each. So, on your question number 6, where you were supposed to cross a heterozygous, two heterozygous individuals, that means they both have the same genotype. They both have a capital letter and a lowercase letter. So look at what you did for your number 6. Do you, across the top, do you have a capital letter and a lowercase letter? Or like I have over here, capital letter and a lowercase letter. But the other individual has to have the same, a capital letter and a lowercase letter. Now, in this combination here, let's go on here. A monohybrid cross means you're just showing a cross for just a single trait. A dihybrid cross means you're doing it for more than one trait at the same time. That gets a lot more complicated. Instead of a box that has a Punnett square that has 4 boxes, it'll have 16 boxes. It's a lot more complicated. And you can imagine probably for any combination you wanted. Here is a Punnett square. Where is this? What would you describe this individual as? How would you describe it? Had a capital letter and lowercase letter, we would call it different. What's the word for different? Hetero, right, okay. So this one is heterozygous for the dominant trait. The dominant trait, no. What did you say? You probably were right and I was wrong.
I said the homozygous.
Yeah, they're homozygous. They're both the same and they're green. Okay? Apparently green is a recessive trait. This one over here is what now? Okay, that one's heterozygous. It has the one dominant trait, and looks like yellow for seed pod is a dominant trait. And when I look at these individual genotypes, what combinations do I get? So when I'm giving the genotype, I always list it in this order. How many of them are homozygous dominant? Are there any in the offspring that are homozygous dominant? Are there any of them that are homozygous dominant? Nope, there's none of them that are capital Y, capital Y. Okay, so we would write down 0 for that. How many of them are heterozygous dominant? How many of them have a capital letter and a little letter? 2, right? So this number would be 2. How many of them are heterozygous recessive? 2 lowercase letters. 2. So the genotype is 0 to 2 for that one right there. Make sense? 0 to 2. Now, phenotype is what it looks like. What do these look like? That's yellow. That's yellow. This will be green. This will be green. So the phenotype, I give the dominant one first. So how many of them have the dominant trait? Those two. So it's 2. How many of them have the recessive trait? 2. So it'd be a 2-to-2 ratio. 2-to-2 ratio. Okay, here's one of those. This has to do with, I think, had to do with corn. And what color are the corn kernels? Like Indian corn has different colors, right? And so that would be kind of like what we would have. If you've ever seen an Indian corn cob, they're not all yellow like they are what we grow typically around here, but you can have different colors in there. But this would be purple, or the other one's yellow. Okay, so yellow is actually, as far as Indian corn, yellow is a recessive trait because we give it a lowercase p. We give a capital P for purple. So here's, this one is, how would you describe this one? They're both capital letters, so what do we call it?
Dominant.
Okay, it's dominant, and what word do we put in front of it? Since they're both letters are the same? Homozygous dominant, good. And then this one would be what? Okay, right, homozygous recessive. So when I put, and they have segregated, notice that these were together, now they're in separate columns. These were together, now they're in separate columns. They segregated. Now we put them together, put the gametes together, and we make what we call a zygote. That's the first cell that we form. And what would be the genotype ratio? Well, it says down here. How many of them are both capital letters? 0, right? How many of them are big letter, little letter? 4 of them are like that. How many of them are little letter, little letter? 0, right? So we get 0, 4, 0. Now, let's go by looks. What will this corn kernel look like? It has one capital P, so it will be purple. What will this one be? Purple, purple, purple. So the dominant trait's purple. So how many purple ones do we have? 4. How many yellow ones do we have? 0. Okay? So I think some of you are starting to get this pretty good, I think. Now, if we took 2 of those and crossed them, so in other words, this would be the 3rd generation now, that we would take the capital P, little p, segregate them, capital P, little p, segregate them, and put them back together to form the gametes. I have 2 capital letters, 1, 2 that have uppercase, lowercase, or heterozygous, 2, 1 lowercase, 1. Now, what do they look like? This one's going to be what color? Purple. This one will be purple. This one is going to be purple. This one's going to be yellow. So how many purples? 3. How many purples? I tricked you, didn't I? I tricked myself even. I didn't even remember what I even asked. How many of them are purple? 3, right? How many are yellow? 1. Okay, good. So what's given up there is what I have written down here, what I've already said. You always write the homozygous dominant one first, then the heterozygous dominant. So in other words, the dominants always come first, and then we end with the homozygous recessive. The recessive one is in this kind of dominance that we're talking about here, is only possible if they're both lowercase or homozygous. Okay, now one of the things that we need to do sometimes when we're working with organisms is to figure out, okay, I have an organism here, but I don't know what its actual genotype is. Let's say it has a dominant trait, but I don't know, is it capital T, or is it capital T, little t? Okay, I don't know which one it is. So what we do is something called a test cross. Okay, in a test cross, we cross it with one that is homozygous recessive. So every test cross, you're crossing whatever it is with a homozygous recessive. Now I did that over here. Let's say we see this tall plant, but we don't know, is it capital T, or capital T, little t?
Trevor has to leave and he says he needs to get something from you. It's spontaneous having to leave. Thank you.
So if I cross it with homozygous recessive, I'm going to get this. So if I see that the offspring are all tall, then the organism in question must have been capital T, capital T. Now, that's how I figured that out. The other possibility, if it's this, notice what I get. I get half of them that are tall, and half of them that are short. So if I find any that are short, that means that the organism in question had to be heterozygous. That's why we call it test cross. But in each case, we're always crossing it with a homozygous recessive to be able to verify which one it is. So those are the different combinations that we had. I just used T's, but here they're using P's. But it's the same idea. And remember, you did the pedigree chart, and we're just trying to figure out the presence or absence of certain traits. In your book, if you turn forward a couple pages, they show the passage of a gene for hemophilia from one generation to the next. Remember, you probably had an elementary, I don't know, maybe you didn't, but you knew of an elementary art teacher that we had, not the one we have right now, that Mrs. Hespy, and she has, I think it's her oldest daughter, is a hemophiliac. But she has what we call sometimes known as bleeder's disease. She has trouble with her blood clotting properly. So she has to have treatment periodically to enhance her blood's ability to clot. You know, if she gets cut or bruised. Those are both bad situations. But in that chart that you have in your book, it traces the hemophilia disease in the royal family in England.